Welcome back, students. Now in this section I'll be talking to you about the biological importance of other amino acids. We finished glycine, serine and selenocysteine, which is got from serine. Now let us look at alanine. What does the name mean to you? What is it used for? Alanine transaminase. Yes, alanine is involved in the formation of pyruvate. Plus it is involved in the inter-organ transport of ammonia in the non-toxic form. Remember the glucose alanine cycle? So that is what is the main significance. So it is involved in the inter-organ transport of nitrogen. The glucose alanine cycle is especially important during starvation. And we have alanine transaminase. We'll go on to threonine. What is threonine useful for? Remember, threonine is a hydroxyl group containing amino acid. So proteins which undergo covalent modification. Where does the covalent modification take place? It has to take place if it has a hydroxyl group. So there are many of these proteins where the threonine residue acts as the site where hydroxyl group gets phosphorylated, and it helps in the covalent modification of different enzymes. So the OH group is the site for phosphorylation. Please also remember that threonine is used for the attachment of carbohydrate residues in glycoproteins. This is regarding the importance of different amino acids. We'll look at methionine. What is it used for? I have kept on telling methionine is activated to form S-adenosyl methionine, and which gives its methyl group to an acceptor to form methylated product. So let us look at this. So it is methionine is involved in transmethylation reactions. The enzymes are transmethylases or methyl transferases. S-adenosyl methionine is converted to S-adenosyl homocysteine. We have a methyl acceptor and a methylated product. Methionine is a precursor of cysteine. Please remember that cysteine becomes essential if methionine is not there. So let us look at how methionine by transmethylation reactions helps in the synthesis of which product. Now these are all the products. Can this be asked in the MCQ? Yes, it can be asked in the MCQ, and hence you should be knowing about these different methylated products. Now one important thing is, first one is guanidoacetic acid. Why have I put it first? Just now I told to you how guanidoacetic acid is formed from glycine. Glycine is the precursor for guanidoacetic acid, and this undergoes methylation to form creatine. Norepinephrine is converted to epinephrine by methylation. So what is the difference? Nor does not contain methyl group. No methyl group. Norepinephrine. Methyl group, epinephrine. Okay, that is how you can remember. Norepinephrine does not have a methyl group. Epinephrine is metabolized further to metanephrine. Norepinephrine is also further metabolized to normetanephrine. Ethanolamine is converted to choline. As I said, serine to ethanolamine, ethanolamine to choline. Choline is an important precursor for acetylcholine. Acetyl serotonin is converted to melatonin. Nicotinamide is converted to N-methyl nicotinamide. tRNA is converted to the methylated tRNA. Carnosine is converted to anaserine. But out of all this, what looks familiar in all these methylated products? If you look at it, most of them are involved some way or the other with the brain, and that most of them are neurotransmitters. One way or the other, either they are neurotransmitters or they are the precursors of neurotransmitters. Epinephrine, normetanephrine, metanephrine, choline to acetylcholine, melatonin, N-methyl nicotinamide, all these are involved in the brain and in the formation of neurotransmitters. Now let us look, methionine is over. Let us look at cysteine, which can be got from methionine. Now cysteine is present in large amounts in keratin of the hair and nails. So your hair and nails contain a lot of sulfur containing amino acid, that is cysteine. What's the role of cysteine? It helps in the formation of disulfide bonds. So cysteine is supposed to explain why some people have got curly hair and why some people have got straight hair. Somebody is having curly hair, there is more of disulfide bond formation between cysteine residues which are far away. More the disulfide bond residues, more is the curliness of the hair, and straight means lesser disulfide bonds. So people nowadays somebody who is having curly will want to have straight, and somebody who is having straight will want to have curly hairs. So all they do is that they help in the use chemicals and see that the disulfide bonds are being formed between cysteine residues. Cysteine is also required for the formation of glutathione. I will keep on telling glutathione is gamma glutamyl cysteine glycine. Cysteine is also required for the formation of taurine. Where have we heard of taurine? Remember taurocholic acid. So it is used for the conjugation of bile acids. Taurine is used for the conjugation of bile acids. Taurocholic acid, taurochenodeoxycholic acid. So are the examples. Cysteine is involved in disulfide bridge formation and stabilizing the protein structure. I have already told to you about it. Now one important thing is cysteine on decarboxylation forms beta-mercaptoethanolamine. Big name, but it is mainly used for CoA synthesis, coenzyme A synthesis. That is the thing to be remembered. Cysteine is required for the formation of coenzyme A. MCQ, what is cysteine used for? Cysteine is used for CoA formation. Cysteine is used for taurine formation. Cysteine is used for glutathione formation. Cysteine is present in hair and nails. Cysteine is required for stabilizing protein structures by forming disulfide. You take insulin. Does it have cysteine? Yes. Cysteine disulfide bridges are present. Whichever you take immunoglobulins. Yes, the protein is stabilized because of disulfide bridge formation. Now let us look at another amino acid, that is glutamic acid. What's the difference between glutamic acid and glutamate? No difference. One is the ionized form and the other is the unionized form. Remember, the ic acid is converted to the 8 when it loses its proton. Glutamic acid is converted to glutamate if it loses its proton. I have been telling about this. There is no difference between glutamate and glutamic acid. But is there a difference between glutamine and glutamic acid? Yes, one extra amino group. Glutamine is converted to glutamate by glutaminase, or it is converted by glutamine is converted to glutamate by glutaminase. So this is the importance of glutamic acid. Let us see what is its use. Where have we heard of glutamic acid? Glutamate, in other words, alpha ketoglutarate to glutamate, transamination. The most important thing is transamination. Now glutamate also forms N-acetyl glutamate, which is a modifier of carbamyl phosphate synthase 1. What is CPS1? CPS1 is the regulatory enzyme of urea cycle. So it forms N-acetyl glutamate. Remember, glutamate is a donor of the nitrogen for the formation in the urea molecule, either as aspartate or as ammonia. Both the things come from glutamate. It also stimulates the urea cycle. So glutamate forms N-acetyl glutamate. Glutamate by itself is a neurotransmitter. It is supposed to be an excitatory neurotransmitter. Glutamate is also involved in glutathione formation. Gamma glutamyl cysteine glycine is glutathione. Glutamate forms GABA. Have you heard of GABA? Yes, you should have been hearing about GABA by now. This is also a neurotransmitter. So glutamate undergoes decarboxylation in the presence of pyridoxal phosphate to form GABA. Now please again note here, amino acid metabolism, principal role is of vitamin B6, pyridoxal phosphate. It forms GABA, gamma amino butyric acid. Now there is one more important role of glutamate in different proteins, and what is that? It is present in blood clotting factors. So what will happen is, when blood clotting is going to take place, there has to be carboxylation. Now in this case, it will be the gamma carboxylation of the glutamate acid residue. So this will help, especially in prothrombin, will help in the clotting process. Vitamin K is also required for this gamma carboxylation of glutamic acid. So the vitamin K dependent clotting factors have glutamic acid residues which undergo gamma carboxylation. So, and this is important in blood clotting. That is the importance of glutamic acid. Now let us look at glutamine. Glutamine Glutamine is used in the synthesis of purines N3 and N9. It contributes N3 and N9 of the purine ring. It is also involved in the synthesis of pyrimidines where it gives N3. So what is it? All this, why am I talking? Because it is in MCQ. Glutamine is used for. It is also used for as a conjugating agent. Phenyl acetyl glutamine. It donates amino groups to amino sugars and amide group to nicotinamide. Where have you heard of nicotinamide? NAD, nicotinamide adenine dinucleotide. It also is involved in buffering, especially during acidosis. It donates its amino group to the buffer and helps especially in the tubular buffering, especially during acidosis. Let us go ahead with aspartic acid. Aspartic acid, main use is in the malate aspartate shuttle. It also contributes, remember, aspartate is given one more amino group to the urea. Synthesis of purines N1 and the 6th amino group. Whole of the aspartate molecule is incorporated into the pyrimidine ring N1, C4, C5, C6. The whole aspartate comes from aspartic acid of the pyrimidine ring. Now let us look at aspargine. Aspargine is mainly in protein biosynthesis and ammonia trapping in the brain. Again, we don't want ammonia toxicity. So the ammonia trapping in the brain also helps decrease the ammonia toxicity in the brain by the formation of aspargine. Lysine. Now lysine and hydroxylysine are important in collagen biosynthesis. Remember, the collagen biosynthesis has to have hydroxylation followed by glycosylation. So this pro-alpha chain of collagen, the lysine residues undergo hydroxylation. So it forms hydroxylysine. So lysine is important. Why is it important? Not only to form hydroxylysine, that will help in the formation of pro-alpha. Further, lysine undergoes deamination to form all-lysine, and all-lysine is important for the cross-linking of the collagen and giving strength to the collagen. The collagenous tissue has more strength because of cross-linking, between the different tropocollagen molecules. So during this, lysine is acted upon by lysine oxidase or lysine hydroxylase. Both these enzymes either before or after will act and help in the cross-linking and giving strength to the collagen molecules. And it is present even in elastin molecules. Lysine is also involved in the formation of SCIF base, what we call as SCIF bases, especially for linking pyridoxal phosphate to proteins. It is important in maintaining structure of hemoglobin and it is present in large amounts in histones. Lysine is one of the positively charged amino acid. What are histones? Histones are proteins which are associated with chromosomes. Now in chromosomes or DNA, there are a lot of nucleotides and all these nucleotides contain phosphate group. Phosphate groups are negatively charged. The compact structure of the chromosome would not have been possible because these negative charges would have repelled each other. So we have certain protein which will see to that these negative charges do not repel each other. At that time, a compact chromosome is possible. So it requires a protein which is rich in positive charges, and lysine is the one which is present in histones and gives that the rich positive charges that are present. So it is present in large amounts in histones. Lysine is also the precursor of carnitine. Carnitine is involved in beta oxidation. Carnitine acyltransferase is the regulatory enzyme. It is involved in the carnitine shuttle wherein acyl-CoA is transported from the cytosol into the mitochondria. So lysine is a precursor of carnitine. So let us go on to the next amino acid, that is arginine. What does the name mean to you? Again, arginine is arginase, urea cycle. That is where it is important. It is involved in the formation of creatine. Remember, the guanidoacetic acid is got from glycine and arginine to form guanidoacetic acid. It is a precursor of nitric oxide. This is one of the most important functions of arginine. Now let us look at another amino acid, histidine. Where is histidine present? In many tissues, wherever positive charges are involved. Plus, one important characteristic feature of histidine is its pKa of the side chain. The imidazole ring has a pKa which is very close to the physiological pH. So, whichever protein contains a lot of histidine residues, it can act as a very good buffer. The buffering capacity of a protein depends upon the number of histidine residues it has got. So, it is involved in the buffering action. It is also involved in the formation of histamine. What is histamine? Histamine, remember, it is involved in mast cell and the production. It is a neurotransmitter, histamine, and it is also involved in 1-carbon metabolism. How is histidine converted to histamine? All is decarboxylation. Remember, all these biogenic amines are got from their precursor amino acid just by decarboxylation. One decarboxylation, histidine is converted to histamine. One decarboxylation, glutamate is converted to GABA. It is also important in tyrosine to tyramine. So all these biogenic amines are got by decarboxylation. So let us look at a brief look on nitric oxide. As I said, it is got from arginine. Arginine, in the presence of nitric oxide synthase, forms citrulline and nitric oxide. Again, we are talking of the biologically important compounds that are found from different amino acid. That's why I am talking about this here. The enzyme is nitric oxide synthase, NOS, what we call as NOS. Now there are three different isoenzymes of nitric oxide synthase. They are called as the inducible NOS, the endothelial NOS or eNOS, and the neuronal NOS or the nNOS. nNOS, eNOS, iNOS. So we just add N here, means it becomes neuronal NOS. If we add E, it becomes endothelial NOS. So what is the function of this nitric oxide synthesis? What are the different functions? To produce nitric oxide. What is the function of nitric oxide? Now, nitric oxide is one of the oxygen-containing free radical. Basically, free radicals, we keep on calling them as villains, but free radicals have a role in the body. Only if they are in excess, you can make them as a villain. So, if they are in the balance, they definitely have a role in the body. Now, nitric oxide discoverer even got the Nobel Prize for the discovery of nitric oxide, and it is considered as the molecule of the 21st century. Now, it is a biological messenger. Its main function is vasodilation. The scientist who showed that nitric oxide is not the endothelium derived relaxing factor, EDRF, which is released from the atria of the heart, is known as nitric oxide. The one who showed it that was the discoverer of nitric oxide. So it is basically a main function is vasodilation. It mainly functions as a vasodilator. It is involved in neurotransmission. It increases the ability of immune system to kill tumor cells or the parasites. So these are the main functions of nitric oxide. It is nitric oxide, and it acts via the formation of cyclic JMP as the second messenger. So we'll talk more about cyclic JMP and its effects as we talk about different other hormones. So right now let us remember that it is a vasodilator. Most important function is vasodilation and its ability to kill tumor cells or parasites. And then now let us talk about phenylalanine. Now, most of the actions of phenylalanine are acted upon by its conversion to tyrosine, phenylalanine hydroxylase. So all the actions of tyrosine are attributed to phenylalanine. Phenylalanine is the essential amino acid. It is present in proteins. So let us look at tyrosine. Tyrosine is involved in the synthesis of melanin, in the synthesis of catecholamines, and the synthesis of thyroxine. Important MCQ point of view and also from the point of clinical point of view. So let us look at the synthesis of catecholamines from phenylalanine or tyrosine. So again we have simple set of reactions. I have just depicted them out here. Since you need not know the reactions, I am just going to tell you what is the carry home message in this. Please remember, phenylalanine to tyrosine enzyme is phenylalanine hydroxylase. It requires tetrahydrobiopterin and dihydrobiopterin is released oxygen. Tyrosine to dopa is a similar reaction. So it is tyrosine hydroxylase. Tetrahydrobiopterin is converted to dihydrobiopterin. Dopa is converted to dopamine by dopa decarboxylase. All the deep carboxylases, remember, require pyridoxal phosphate, and dopamine is not only a neurotransmitter, it is somewhat similar to a biogenic amine that is formed from histidine. Histidine to histamine, tyrosine to tyramine, dopa to dopamine. All these are catalyzed by decarboxylases. Now we'll go further. Other than dopamine is formed, one neurotransmitter. Which are the other catecholamines? Other catecholamines include norepinephrine and epinephrine. So dopamine beta hydroxylase converts dopamine to norepinephrine. What is important here? Vitamin C, oxygen and copper are important. So for the synthesis of catecholamines, we require phenylalanine, we require tetrahydrobiopterin, we require vitamin C, and for the synthesis of epinephrine, we also require S-adenosyl homocysteine. Okay, so S-adenosyl methionine, which is converted to S-adenosyl homocysteine. So basically, vitamins are required, tetrahydrobiopterin is required, methionine is required, phenylalanine is required for the synthesis of either dopamine or norepinephrine or epinephrine. This is an important reaction. Please note that vitamin C is required for dopamine beta hydroxylase. So let us look at the other amino acids as we go ahead. I'll talk more about the other amino acids in the next sessions and the biological important compounds formed from other amino acids in the next session. Thank you.